Okay, hello everyone.
Hello, Miss Nandi.
Okay,
Hey,
so hmm?
Hi.
Hi. Okay, so let's start. So chapter 12 today. Okay. So um um do you remember that Dolly the sheep? Is anybody remember Dolly the sheep? Hmm?
not really. No.
Not really? Okay. I think it's yeah. Okay, so my pencil. Yeah. So Dolly the sheep first cloned mammals. Now you remember? No? Okay. I forgot the year, but no, I think I remember. It's
Yeah,
1996.
I remember Dolly now.
Yeah, yeah. Oh my God, my pencil. I cannot see my pencil. Okay. So it was the year 1996, University of Edinburgh. Edinburgh, at Scotland. They successfully actually cloned Dolly. Okay. So that Dolly, how actually they made Dolly, it's a history. So Dolly had three mothers. Okay. So you can see the picture of Dolly here. That's our little Dolly. Okay. And she had three mothers. This one, and this one. So what actually they did um, first from this white sheep, Scottish white sheep, they get the other cells. Okay, so from the other cells they take out only the nucleus, means the DNA. Okay, and from the second mother, she was black-faced sheep, Scottish sheep. She donated one egg cell. Okay, but the scientists, they take out the nucleus from that egg cell, so they only used the cytoplasm of the egg. Okay, now they inserted the egg from these cells, from the other cells of this mother, and they inserted that nucleus inside these egg cells. Okay, and then they actually induced the egg in vitro condition. Means they applied heat, electricity, so that this nucleus and cytoplasm together, they get fused, and as a result these two started working as a zygote. Okay, and then they after when that zygote started dividing, and ultimately it become blastocyst. Blastocysts means 16 cells were there. It's a ball of 16 cells. Then they inserted the blastocyst inside this surrogate mother. Okay, and then finally after 5 months, this surrogate mother gave birth to to this Dolly. So over here, this fusion of the cytoplasm and the DNA, or you can say that the nucleus of another cells, this fusion of these two cells, they actually successfully they fused 277 cell fusion they did, scientists. And there were 13 surrogate mother. Okay. Nobody actually was successful to give birth. Only one mother successfully gave birth to Dolly. So over here, this is an example of biotechnology. Okay. So when we use living systems or living organisms in the medical, industrial, or in any technological applications, that is called biotechnology. Okay. So in this chapter, we are actually going to study the different applications of biotechnology. Very interesting chapters. You can see here what we did. We grabbed only the nucleus portion from one cell, okay, and the cytoplasm of other cell, and ultimately we combine these two together by using technology, okay, and we convert these two cells into zygote, and this is an example of genetic engineering. So when we apply the principles of engineering into the bio or medical sciences, that's the genetic engineering. Genetic engineering is just an example of application of biotechnology. So in this chapter, we are going to study about different applications of biotechnology and how we use different tools. So terminology: biotechnology, manipulation of living organisms or their components to produce useful commercial products. So basically, when we manipulate any systems or any living organisms to make a recombinant DNA, because we want a desirable trait to make, okay, then that process is called biotechnology. Genetic engineering: techniques used to cut up and join together genetic material from different species and to introduce the result into an organism in order to change one or more of its characters. So basically, purpose over here is to make desirable traits. Okay, like we don't want to tomato to be ripened. Okay, we want longer shelf life for the tomatoes. So and also we don't want the seed inside the tomatoes. So what we are going to do by using genetic engineering, we actually can make the tomatoes that will not ripen. Means it will ripen ultimately, but it will stay fresh or green for long period of time. Okay, we and even we can produce tomato without the seeds. Okay, so to do that, what we can do, like let's say that tomato, we want really large red tomatoes. Okay, now we can take out the nucleus or the genetic material from these tomatoes, and there is another species of tomato which is not so tasty, but it can stay fresh for a long period of time. So what we can do, we can cut the gene or DNA from this tomato, and we can insert into this one. Okay. Similarly, if there is another species or another variety that does not have any seeds, we can cut the piece of genetic material, small piece of genetic material, and we can insert into it. Okay, so basically this tomato, so I talked about 3 different species, 3 different varieties of tomatoes over here. This one, it's really large and tasty, taste good. Okay. This this one, it's shelf life longer shelf life. Okay. And this one, it does not have any seeds. Okay. So we chose the large one. This is tasty. We preferred this tomato over these two. Okay. But these are the good characters that we also like. Okay. So what we can do nowadays, then we can grab a little bit of genetic material from these two tomatoes, and we can insert into this large tasty one. Okay. And we can get all these three qualities into just one, within just one tomatoes. So what we did, we actually manipulated the genetic material, the DNA. We know the genetic map of almost all organism. Okay. So let's say this DNA belongs to this tomato. Okay. And this is the portion of gene that is responsible for longer shelf life. So what we can do, we can just give a cut over here and here, and we can get this portion out of this chromosome, and then we can insert this chromosome with this DNA. Okay, we can do that. And this process is called genetic engineering. So this tomato has its own DNA, right? When we added this small new portion of chromosome from this tomato, then this chromosome is called the recombinant DNA, or and this it is called recombinant DNA or recombinant chromosome, because this is the original portion of DNA for this tomato, and this is the extra portion from here to here that we got from this tomato. So this chromosome, it's completely new type. Okay. So that is why, and it is carrying basically two different types of chromosomes that came from two different species. So that is why it is called recombinant DNA. And this process is called recombinant DNA technology. Okay. So recombinant DNA technology, insertion or modification of genes to produce desired proteins. So this genetic engineering and to do the genetic engineering, we actually need to make recombinant DNA. Okay. So for genetic engineering, this is the tool, that recombinant DNA technology is a tool for the purpose of genetic engineering. And this genetic engineering is one application of biotechnology. Okay. So I hope you got the relationship between this biotechnology and genetic engineering. So recombinant DNA technology, another example nowadays, like we make growth hormone, we make insulin hormone, human insulin hormone. Previously, for the diabetic patients, you know what we used to do? We used to collect insulin hormone from sheep, mostly from sheep's body. Okay. And then we used to inject that insulin inside human, whoever that needed, because that insulin actually help extra sugar to metabolize. Okay. So that's why that insulin hormone is very important. And the people who cannot make enough insulin, we used to inject that extra insulin hormone from sheep's body, and it had really bad effects, because that sheep insulin, not all people, but some of the people used to have, allergic reactions and all those. Now what we do, we actually get a small piece of insulin gene from our chromosome, means from human chromosome, human DNA, we get that insulin gene. Okay, and then we insert that insulin gene inside a bacteria, and we allow bacteria to divide, to multiply. Okay, and then from those bacteria's cells, we actually induce the bacteria so that that gene actually expressed, and inside the bacterial body, that gene can make insulin hormone, and then we do the centrifugation, and we actually purify the insulin hormone from the bacterial body, and that is the hemulin. It's actually human insulin, and it does not have any allergic effect or any adverse effect on human. Okay, so how we actually do, we use recombinant DNA technology. Now to do that recombinant DNA technology, there are lots of bacteria who carry special type of enzymes, you can say. Okay, and they have special activity, they can cut the DNA. So we use those bacterial enzymes or bacterial protein to cut our insulin gene. So what we do, we cut the gene, like if it is a human gene, human chromosome, we just cut out the only portion, that is responsible for insulin protein. So we cut that portion. Okay, at the same time, we cut a small portion of plasmid. Plasmid, the bacteria, they have extra chromosome. So those are the plasmid. So we give a cut inside that plasmid, and then we insert that human chromosome inside that plasmid. Okay, so now it's a recombinant DNA, recombinant chromosome. It's a completely new type of chromosome. Previously, this was the original plasmid, original DNA. We have changed it by inserting a human DNA portion into it. And now at this moment, this plasmid, it is actually carrying two different type of chromosome. One type that came from the bacterial plasmid, and the small portion that actually came from human. So it's a human chromosome or human gene. So it is the combination of both human and bacterial chromosome. That is why it is a recombinant DNA, and this process is called recombinant DNA technology. Now what we do, we actually keep lots of this type of plasmid, recombinant DNA plasmid, along with the bacteria. So inside the let's say this is that agar plate, agar media, solid media. So when we inoculate this media with the bacteria, we also add this recombinant plasmid inside that media. Bacteria automatically actually pick up all those plasmids. Okay, and when bacteria divides, it also plasmid also is going to divide inside the bacterial body. So as a result, bacteria help us to make multiple copy of this small piece of human insulin gene. Okay, so from one bacteria, or you can say from just one small piece of human insulin chromosome, we can make multiple human insulin gene. Okay, now I hope you remember that what happens. So from DNA, that transcription occurs. Then after transcription, that's the translation. Okay, during translation means it is called the gene expression. When gene is going to be expressed, means it is going to make protein. Okay, so we induce that gene expression over here by adding electricity or by creating magnetic field, sometimes different type of acids or different chemicals material. Okay, and when we induce the bacteria, then bacterial that small plasmid, they start making protein. Okay, so basically inside all the bacteria, that insulin gene is going to make that insulin protein. Then we collect all the bacteria, and then we actually purify, we do centrifugation and all those, and then we can get that insulin hormone. So this whole process is an example of recombinant DNA technology. And this is actually one tool of genetic engineering. Okay, so why it is genetic engineering? Because we are manipulating the DNA or gene or the chromosomes of different organisms. Okay, and so basically two things are here. We are manipulating the genetic material, DNA material, and at the same time we are applying the principle of engineering. Okay, so together that's the field of genetic engineering, and recombinant DNA technology is just one example or one tool. So tools of genetic engineering. So the deliberate modification of an organism's genetic information by directly changing the sequence of nucleic acids in its genome is called that genetic engineering. So deliberate modification. Okay, so over here, yes, we actually deliberately changed the chromosome arrangement of this plasmid. Okay, maybe at this moment this plasmid had 50 genes. Okay, now once we cut out this portion and we added human insulin genes, maybe now it has 52 genes. Okay, or maybe 49 genes. That can happen. So of course this combination and these combinations are completely different from each other, and we deliberately did it to produce insulin hormone. Okay, so that's the genetic engineering. Nowadays we make growth hormone, so many things we nowadays we make. Okay, all the vaccines, different types of research enzymes, proteins, so by using this recombinant DNA technology. So major tool is recombinant DNA technology. Recombinant means DNA joined to other unrelated foreign DNA. So what we do, we actually cut a gene portion of DNA, okay, and insert that portion into another DNA strand. To cut a DNA, we use special type of enzymes, and these enzymes comes from the bacteria, and they are called restriction endonucleases. Okay, so this special type of enzyme, it actually comes from another bacteria. We use this endonucleases enzyme as a scissor. You can say that they are actually molecular scissor. Okay, so we use to cut the DNA of human and also the plasmid. Okay, so these restriction enzymes are very, very important for this genetic engineering purpose. The recombinant DNA is then introduced into a host organism host organism over here, that's the bacteria. If the introduced DNA comes from a different species, the host organism is now called transgenic. Okay, so over here, when we make the insulin, so bacteria, they are the host. Okay, and this plasmid who is actually carrying that human chromosome, they are called vector. Vector means who carry a specific chromosome. And over here in this recombinant plasmid, this portion of the genes that came from the human. So human and bacteria, completely two different organisms. Okay, so now this plasmid, you can say it's a transgenic variety. Okay, so basically completely new type, and it's a combination of two two chromosomes or two DNA or two genes that actually came out from completely two different organisms. So goals of genetic engineering: create organisms that synthesize products human need, like insulin, eliminate undesirable phenotype traits, suppression of ripening in tomatoes, like we don't want seeds. Okay, we want longer solve life. Okay, um, um, we can choose the color, we can choose the size, so many different things. We don't when we want, we apply it on plant. We don't want any weed, or you want the plant to be weed resistant. Okay, we want the plant to be any pest resistant, so we don't have to. Nowadays we are making this weed resistant and pesticide resistant or pest resistant plant by using this genetic engineering. So we really do not have to apply any pesticides or any chemicals, and so that it will not have any adverse effect on us, on our body. So that's the goals of genetic engineering. These are the so many different types of tools of biotechnology that we use. Okay, like gel electrophoresis. Okay, we can identify who is the victim, who is the murderer. Okay, we can go behind a specific DNA molecule, particular DNA molecule. We can find out the paternity. Polymerase chain reaction. So at the murder site, we have just a small portion of hair. Okay, by using that small hair, we can make millions of hair sample. Okay, so that's the polymerase chain reaction. So all these things are the tools of biotechnology. You have to study all these tools from the text. I will just go over a few examples. So restriction enzymes or restriction endonucleases. I told you that it comes from the bacteria. So basically what happens for the bacteria, bacteria, some bacteria, not all, they actually carry this restriction enzyme. It's their protective device. Okay, so what they do when they get infected with virus, they actually use these restriction enzymes to cut the viral DNA. Okay, so they use this restriction enzyme to cut the viral DNA into small pieces. As a result, the virus cannot multiply inside the bacteria. Okay, so a scientist observed this mechanism, and now we actually use that enzyme to cut our any human, any DNA chromosome, any chromosome from any organism. Okay. So this restriction enzyme, or it's also called restriction endonucleases. This enzyme, they actually recognize and cut specific fragments. So basically what happens, like for E. coli, E. coli can recognize these segments, GAATTC. Okay, so wherever, let's say this is a chromosome, okay, human chromosome, that E. coli has endonuclease enzyme. So wherever, let's say GATTC, 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 okay? So DNA double stranded, right? CTTAAG. So over here we should have, one more A over here. So CTTAAG. CTTAAG. So that's the DNA double helix of human chromosome. So this EcoR, the name of the restriction enzyme is EcoR1, that comes from bacteria Escherichia coli. Okay, that restriction enzyme, wherever it will see this sequence GAATTC inside the human chromosome, it actually gives a cut after. So it will give a cut in between G and A, G and A, G and A. Okay, so look, this side it's GAATTC, this side GAATTC, so it will cut in between G and A, G and A, G and A. So basically it gives a cut like this. Okay, now so if you see, it comes like G C, if this portion GCTTAA, GCTTAA. Okay, so this is one portion. Now this portion GCTTAA. So GCTTAA. So they can actually recognize a specific sequence of nucleotide. So this sequence is very specific with EcoRI. Another endonuclease, maybe that comes from Bacillus bacteria, that sequence should be completely different. So this sequence is very specific with this specific endonuclease enzyme. So by seeing this small piece of DNA fragment, we can recognize that which enzyme actually cut these DNA pieces. Okay, now from here, I hope you have seen that GAATTC. So if you read it forward and backward, it's the same. Okay, so it's called palindromic sequence, and this restriction endonuclease, this restriction enzyme, they actually recognize this palindromic sequence, and that's how they cut both the DNA strand. And where they actually give a cut on the DNA strand, that site is called a recognition site. So this GAATTC, the small piece inside the DNA, that is the recognition site, one of the recognition sites for this EcoRI. So the name of the endonuclease comes from this first three letter comes from the bacteria. R, that is a very specific cysteine of this bacteria, and this is the first enzyme we collected from E. coli bacteria, so that is why it is 1. So EcoR1. Okay. So recognition sites, DNA is cut on a specific palindromic sequence, typically between 4 to 6 base pairs in length. Over here we had 1, 2, 3, 4, 5, 6 base pairs for EcoRI. Different bacteria produce many different restriction enzymes that cut genetic material at different sites. Okay, now I told you that this is the recognition sites or palindromic sequence for the EcoRI. So wherever they are going to see this sequence, they will give a cut. They will not look that it is whether this piece of DNA came from human or from bacteria or from virus or sheep or goat. They will not going to look for that. Wherever any small piece of DNA, and if there is this recognition site, they can see, they will give a cut. Okay, so over here, when we made that, insulin recombinant DNA technology, so what we did, we used same restriction enzyme to cut the human chromosome and to cut the plasmid. Okay, so it has a same sequence over here, that could be complementary with these two ends. Okay, so it can be easily fit inside that plasmid. So that's actually the science we want to apply. Like over here you can see, look, if this is one segment, okay, this is one, let's say it came from the plasmid and it came from the human. Okay, so if in a pot inside a plate, if you keep these two, because of the complementarity, okay, so these two strands will very quickly, they will make a double-stranded molecule. They will add up. Okay, they will make small hydrogen bond in between very quickly, and they will be double-stranded again. So that's the complementarity. And this proportion, you can see that the small stretch, a small wing, that is actually unpaired with unpaired DNA sequence. This is called overhang portion of this DNA strand. Okay, so this is the DNA strand from AATTC, and G, this part is called the overhang part, or you can say the small wing of DNA, or small stretch of DNA segments. So this restriction enzyme, one way to cut this DNA sequence is they actually left a small overhang portion with the DNA. And that has a purpose, because if one overhang portion is there, means over here we really do not have any nitrogenous base. So but C goes with G, so it is hanging with G, so T can quickly add up with A, and it's pretty easy to make hydrogen bonds in between T and A and T or G or C. Okay, so and this is one type of cut. Okay, and this type of cut is called staggered cut. So over here, this one, This came from Banhi. Okay, another bacteria. This is actually Bacillus, but this is not Subtilis. I forgot the species, but the bacteria Bacillus. They recognize CCTAGG. This is the portion they can recognize, and they give a cut in between 2G. So in between 2G, they will give a cut over here, one side, and it's palindrome, so this side. So GGATCC, so GGATCC. So that's the palindrome. Okay, so I gave you here one example of palindromes. So no de ba bob abidon. So no de ba bob abidon. So you, whichever the direction you read the same thing. Okay, you go forward or you go backward, you are going to read the same thing, and that is called palindrome, or you can say palindrome sequence. Okay, so that BAMH1, this restriction enzyme, GGATCC, that's the recognition site. Okay, and in between 2G, it gives a cut. So it basically give a cut like this. Okay, that's the cut. So what happened? So GCCTAG, this portion remains together, and GCCTAG, these portions remain together. So this is the overhang portion for this part of the chromosome, and this is the overhang portion for this part of the chromosome. And when, so basically this cut portion looks like this, and this portion, these ends are called sticky ends. Why they're sticky ends? Because they are complementary to each other. It's pretty easy to make hydrogen bond in between. So that's why this type of cut is called that they make sticky ends. These are the sticky ends. Very quickly get attached with each other. Another big bacteria like Haemophilus HAE3, Haemophilus 3, these bacteria, it actually gives a straightforward cut. So recognition sequence is CCGG, okay, GGCC. So in between G and C, they give a cut. So in this case, so we get one top part and one bottom part. Okay, and they it has, there is no stretch or overhang with unpaired DNA sequence, and this type of cut is called blunt end cut. Okay, so restriction enzymes, so there are two different types of cuts. One, that's the sticky end cut or staggered cutting, and another one is blunt end cutting. So blunt ends, staggered cut, sticky ends, and the pieces of DNA produced are called restriction fragments. So this was the whole DNA after cutting. So this piece and this piece are called restriction fragments. So this is one restriction fragments. This is another restriction fragments. The complementary sticky ends can easily anneal. That anneal or annihilation means forming hydrogen bonds. Okay, so when complementary DNA strands, they make hydrogen bond in between, that process is called annihilation. Okay, or annealing. So this staggered cut, when they make hydrogen bond, that sticky ends is much more efficient than the blunt end, because blunt end, they don't have unpaired DNA nucleotides, so it's get a little difficult to add up to blunt end cut DNA pieces than the stagger cut. In both cases, ligase enzyme rejoins the two sugar phosphate backbones through covalent bond. If we keep one blunt end and one staggered cut end, anyway, ligase enzyme, just like we have studied, right, during DNA replication, that if there is any nitrogenous base are there, and that ligase enzyme actually join the two sugar phosphate backbones. So that's how we can join the two restriction fragments together. Now the cloning. Cloning means making multiple of or millions of identical DNA segments. So you have a small piece of DNA, okay? Like let's say it's a small hair sample. By using genetic engineering or biotechnology, you can make multiple copies of this hair, exact DNA copies of this. Okay, so these 1,000 copies will be identical of the small original DNA piece. Okay, so when you do that, when you make the multiple copies of multiple identical copies of one small DNA piece, that process is called cloning. Okay, so from organism also, like from one tomato, when you make 1,000 tomato, that is also cloning. Okay, from one plant, when you make multiple plant, that is also called cloning. So after restriction digestion, so restriction digestion means when we cut a DNA piece with that restriction endonuclease enzyme, means we make a restriction fragments. That process of cutting is also called restriction digestion. We also use this word, the digestion. So instead of cutting the DNA, we can call that after the digestion of the DNA. Okay. So after restriction digestion, genes of interest are incorporated into a vector. Usually we use plasmid or virus. I showed you one example of making insulin where we used bacterial plasmid as a vector. Vector means who actually carry the recombinant gene or the new, or you can say our desired piece of gene who carry, that's the vector. Nowadays we also use virus as a vector. So vector insert DNA into a cloning host. We use bacteria or yeast. Each transformed cell will divide to form millions of cells. This process yields multiple identical clones of the original recombinant molecule. Whatever you have studied here when we worked on the recombinant DNA technology for insulin making. So plasmid, over here we used plasmid as a vector. Okay, bacteria, that's the host. Okay, we nowadays we use virus as a vector. So basically we insert that DNA piece inside the virus's body, and then we can inject that virus inside the bacteria. Okay, bacteria serve as a very good host. Nowadays we also use yeast as a host. So host actually help us to multiply that specific piece of DNA. Okay, same procedure. So over here, we use that cloning. So nucleus, this is the human, any mammal, microorganism or plant, anybody's chromosome, we first take out a small piece of chromosome or small piece of DNA. Okay, then we insert that inside the vector's body. This is the plasmid over here, that's the vector. Okay, Now we insert the vector inside a host's body. Okay, we don't insert, we actually, this is the process of induction, or we actually induce so that bacteria can grab that. I hope you remember the transformation. By the process of transformation, bacteria will grab that plasmid from the environment. B-sub is pretty quick. They pick up very quickly from the environment. We really do not need to apply any induction, or we don't have to induce the bacteria, but for other bacteria we really need to apply a little bit of electricity or heat to induce. Now when host will multiply, that plasmid also will multiply, and so from one bacteria we can get multiple copy of that chromosome. So nowadays we can make different type of protein in the pharmaceutical, for the pharmaceutical companies, like insulin, human growth hormones, different type of vaccines like hepatitis B. Okay, we can alter the organisms with economically useful traits like pest resistance, herbicides resistance, improved nutritional value. So we need more vitamin C inside a potato. Okay, so potato it as such, we get a little bit of potassium. Other than that, it does not have any nutritional value, but it's cheap, right? And its production is pretty easy. Okay, and it does not cost that much. So in future you can see the potato with vitamin C, because we all need vitamin C. It's very important. Or maybe vitamin B complex, okay, or vitamin D, you never know. So we can do that. Okay, so that's another purpose, a source of DNA for study, for the research purposes, for gene regulation, nucleotide sequencing. We actually use this cloning process, and which is genetic engineering. So vectors, remember I told who actually carry the recombinant, not recombinant, who actually carry the foreign chromosome, they are the vectors. Okay, like plasmid, bacterial plasmid, virus, they are the vectors. Now vectors, so is anybody become the vectors? No. To be a good vector, so there are few characteristics. Okay, basically there are three important characters that must be there inside a vector. Okay, so first one is origin of replication. So origin of replication, remember that when we studied DNA DNA replication, I hope you remember that there should be a origin of replication, right? From where actually that DNA replication starts. So inside a plasmid, if this is a plasmid, there must be a origin of replication. Means origin of replication means that specific sequence, okay, from where that DNA replication can start. Okay, so remember that bacteria inside bacterial body, the plasmids, they are self-regulated. Okay, or you can say auto-regulated. Bacterial chromosome, bacteria divides bacterial chromosomes, but plasmid, they are auto-regulated. They actually, when bacteria divides, they divide by themselves, maybe be a little before the bacterial cell division. Okay, so to divide, plasmid must have that origin of replication, otherwise DNA cannot divide. So that is very important point. Okay, somewhere on the vector an origin of replication must be there. Okay, so that it would be replicated by DNA polymerase. Otherwise DNA polymerase cannot start replicating the DNA. there must be a selectable marker, that's the second characters, okay, containing gene that confers drug resistance to their cloning host so that bacterial cells can be grown on drug containing media. Okay, so following the uptake of vector by host cell, one must be able to discriminate between cells that successfully obtained vector from those that did not. So basically what happened, that selectable marker. So inside this plasmid, there should be a portion. So origin of replication is there. Along with the origin of replication, there must be one small portion that should carry one antibiotic resistant genes or any drug resistant genes. Let's say over here, this chromosome, this gene is ampicillin resistant gene. Okay, now what happened? So we are, let me go back over there, then you, okay, so what happened, let's say over here, it has, this is the ampicillin resistant gene, this portion is ampicillin resistant genes. Okay, now we are going to induce the bacteria so that bacteria can take up this plasmid from the environment. Okay, so and after that we allow that those bacteria to multiply. So let's say at this moment we have 100 bacteria. Okay, so from one bacteria we got 100 bacteria, but do you think that all of these 100 bacteria can take up that plasmid? No, maybe out of 100 after induction, maybe 70 bacteria will pick up that plasmid. So 70 bacteria will carry this recombinant DNA. Other 30 bacteria, they will not. Okay, so how are we going to differentiate between this 30 and this 70? So 70 with recombinant DNA, 30 without recombinant DNA. Okay, so there is we should not spend money for this 30 because you know we cannot use this because they are not carrying this recombinant DNA. Okay, so we just need only 70. So how are we going to differentiate? To do that, what we have to do, what we will do, once this bacteria will grow, we will collect all the bacteria from the plate and then we will allow them to grow inside one antibiotic resistant media. So we will transfer the bacteria inside another growth media. It's a growth media. Okay, that growth media will carry ampicillin. Okay, antibiotic. So these bacteria, all these 70 bacteria, they are carrying this plasmid, means they have that ampicillin resistant genes. So all these bacteria will survive here. But these 30 bacteria, they will die because they do not have this recombinant DNA. Means they do not, if they don't pick up this plasmid, that means they don't carry that ampicillin resistant genes, so they will not survive. Ampicillin will kill those. But these 70 bacteria, they will survive because they have that small piece of antibiotic resistant genes that is inserted inside the plasmid. Okay, so this differentiation is very important, okay, economically. So to do that, we must have one selectable marker, so that antibiotic resistant genes that work as a marker. Okay, we can identify which bacteria is carrying the recombinant DNA and which bacteria is not carrying recombinant DNA. Okay, so keeping that marker is very important. So we actually we insert inside the plasmid, we actually insert that small portion of antibiotic resistant genes. Then the third one is the polylinker or multi-cloning sites. Okay, so all these steps, like from here to here, it takes time and it costs a huge amount of money. So when we are inserting this insulin gene, at the same time we actually insert multiple of different type of genes inside the plasmid can carry at the max 50, 55 or maybe 60 genes. Okay, so we actually what we do to save the money and time, along with just the insulin gene, we can insert growth hormone, we can insert gene for hepatitis B, we can insert genes for maybe golden hair. So all those different traits, genes for different traits that we want. Okay, we can insert so that we can save money and time, and that site is called polylinker or multi-cloning site. Okay, so multiple unique restriction enzyme recognition sites that are used for inserting DNA inside the plasmid after restriction enzyme, and all those gene is actually we cut or we digested all these genes with different restriction enzymes bacteria, so their combination is different or nucleotide sequence are different. Okay, and but that specific site over here, you can see this is the polylinker site. These polylinkers are multiple cloning sites. We have lexizines, so many, 1 2 3 4 5 6 7 8 different restriction enzymes we used. So we have 8 different cloning factors over here. We want to make 8 different proteins. Okay, and 8 different types of genes are here. We can insert 8 different genes here by using these are the endonucleases. Okay, who the molecular scissor who are actually going to cut artificially the portion of genes. So these are the three. You can see this is very popular, that PAC19 vector. It's at this moment this is the highest selling vector in the market. Even you can order it at Amazon. Okay, you can. It's very cheap. It's not costly, and if you are Prime member, you are going to get delivery within 2 days. This PAC19 plasmid. So over here, this PAC19, it must have one origin of replication somewhere. Okay, I think this one should be, this area should be origin of replication. Okay, this is the ampicillin resistant genes, and this is the polylinker site for so many different types of protein. Okay, now this polylinker or multi-cloning sites again, look, so over here we have inserted 8 different genes. Okay, now so 2 things are working. Okay, one, so this is the whole plasmid. How do we know that bacteria actually picked up this plasmid? So this is plasmid, and this is the bacteria. Okay, so how do we know that bacteria picked up that plasmid? So that's the reason we have that ampicillin resistant genes here. Okay, to know that bacteria, so basically this plasmid is inside the bacterial body. Now the second problem is, how do we know that all 8 genes, like all these polylinker thingy, are here inside the plasmid? That's the second question. To solve that problem, what you do over here, all these polylinker chromosome like insulin and all those, what you do over here, we have a special marker or molecular site, okay, and that is called reporter gene. Okay, so first what we do, we actually insert a reporter gene over here. So that's the site of reporter gene. Okay, now this reporter gene, it reports something. Okay, so what it reports? It reports that all these things are there or not. Okay, so plasmid picked up all these chromosomes, all these portions of genes or not. So to do that, so what's the characteristics of reporter gene? Reporter gene actually express one specific protein. Okay, usually we use LAGZ. That's one reporter gene. Now LAGZ gene, so once if we treat the bacteria, okay, so let's say that this bacteria picked up that plasmid, and over here we have that reporter genes. Okay, now if we treat this bacteria with another chemical, it's called XGAL. If we treat the bacteria in XGAL, usually LAGZ gene along with the XGAL, it produce one blue color. Basically one pigment they produce that gives the bacteria blue color. Okay, all the bacteria will be blue if LAGZ gene is present, and if we treat the bacteria in XGAL. Now, if this reporter gene is carrying this polylinker site, okay, then it got disturbed. Then it cannot be expressed. So it cannot make any protein. So it can't be expressed. So if this, so basically reporter gene is disturbed and it cannot be expressed. So if this is the situation, then it cannot work with the XGAL. Okay, and as a result, instead of blue color, all the bacteria will be white color because there is no blue pigment is produced because LAGZ is not working. Okay, so when we treat the bacteria with XGAL and all the bacteria is white in color, then we know that polylinker site is nicely inserted inside the plasmid, because reporter gene is disturbed, because that area, that protein or that region is actually occupied by the multi or polylinker sites. Okay, so that's another marker. So these are the three characters. So one, origin of replication, ORI. Okay, then drug resistant genes, and one, polylinker site. And just remember that polylinker sites remain inserted inside the reporter gene. And the process, it's called transformation. Bacteria take up the free DNA from their surrounding. B sub is naturally competent. But for other bacteria, we have to give chemical or electrical shock or magnetic field, all those. Another tool, defined tool, DNA gel electrophoresis. So gel electrophoresis separates the mixed DNA fragments on the basis of their charge and site. So murder site, okay? We have to find out the victim, and from the spot of the murder or from that area, we find out 5 DNA sample. Okay, how are we going to identify and that who is the who actually did murder? So like or let's say it's a rape case. Okay, so from the body of that woman, we found out one DNA, but on the spot we got 5 DNA. So we know that this DNA is the DNA of the rapist. Okay, but on that spot 5 people went. So how are we going to match this DNA with um, uh, this sample? So this is the process of DNA gel electrophoresis. We can actually separate out DNA segments based on their charge and on their size. Okay, any not only DNA, even we can separate out the RNA segments. So any molecular any molecules, actually fragments of molecules, we can separate from the mixed segment based on their charge and size. The charged macromolecules migrate across the span of gel because they are placed in an electrical field. So we actually, if this is the plate, it's a plate. Okay, we use agarose gel, almost like the plate you use in lab, that agarose gel, that solid media. It's yeah, almost same. So we use that one, that agarose gel. Inside the agarose gel, that matrix, you can say the matrix, it has pore. Okay, so the DNA or the small RNA fragments, they actually can move through this pore. Okay, and DNA, it has negative charge. Okay, so what they do, if we add to electrical fields, okay, if we can create electrical currents inside this agarose gel, so DNA, they are negatively charged, so they actually move through these small pores towards the positively charged side. And that's how, and when they start moving through that gel matrix pore, what happened, that large or heavy weight particles, they cannot move faster. So the lighter particles, they move fast, and the heavy particles, they move slow. And we have one ladder, we use one ladder, or one row of known particles. So basically this chromosome we will run over here, and we call that line as ladder. Okay, so we know that who actually raped, this is his chromosome has this much weight, and after the agarose gel, it actually moved from here to here. So the person whose chromosome actually match with the ladder, so we know that that's the victim over here. So that's the whole process. Okay, so DNA molecule has a negative charge, moves towards the positive pole, smaller fragment moves faster, and we can compare the degree of genetic similarities. Okay, so that's the DNA gel electrophoresis. So this is the process. I can show you the picture over here. Okay, so this is the plate. What we do, we actually pour the liquid gel over here. This is the tray or plate. We pour the liquid gel over here, and these are the comb. Okay, you can see that it has teeth. So when this liquid gel is, if you allow it to solidify, it takes another 30 minutes, and that liquid gel converts into it actually solidify. Okay, so when it is semi-solid, what we do, we insert the comb inside that gel, so and allow the gel to solidify. Okay, when it becomes solid, then we actually take out the comb from the gel, and as a result we create small hole or you can say oil inside that gel plate. You can see here. So this is the agarose gel liquid solution. With that, we use one buffer. Buffer actually helps to move the electric charge, electric current. It helps to create one electric current. So mixture of agarose gel and the buffer solution we pour over here, and then we insert that comb and we allow the solution to dry out. So, and after when it's solidified, we take out the comb. So inside the gel, we have a small well, small place basically, where we with a small pipette, molecular pipette, we actually inject the DNA or the sample RNA. One, we keep it as ladder. So let's say we have 5 victims, so we have to make actually 6 oils over here. Okay, so one we will use for the known weight, okay, known DNA particle, and all other oils we will use for the suspects. Okay, so we will inject that, then we will make a electrical field. We will connect it with the electrodes, positive and negative. So this side, the oil side, we will connect the oil side with the negative electrode, and other should be positive. DNA is negatively charged, so it will move, start running towards the positive. So you can see that through the matrix pore, they started moving. So this is the DNA ladder, and we know the size, okay, size of the DNA. Now what we can do, we can match that which one is matching with our known one. And then so when basically when we actually insert the DNA, we use another buffer. Basically we give the color to the DNA, fluorescent color. Okay, and after few hours of this running this gel, then we take out the electrical current, and we can place that agarose solid gel underneath the fluorescent light, and we can see that how it flourishes, and then we can match which one is going with our known one with the ladder, and that's how we can identify. Okay, and this is the way we can even find out the paternity, how the DNA match. Nowadays it's a fashion, we do, okay, so I want to match DNA with my dad or mom, so this is the process. So this is the gel electrophoresis. So I already talked about it. The next tool is polymerase chain reaction molecular Xerox machine for DNA. So we have only one small DNA sample. Okay, we have to do so many tests. Okay, DNA, RNA, so many tests we have to do, and we have just one small sample. So what we do, we actually we clone that DNA sample. We make multiple copies of these, and there is a machine to do that. That machine, that process is actually called polymerase chain reaction. Okay, so we can make multiple copies, identical copies of one gene, several genes, lots of genes. You can just basically in vitro condition, we can make lots and lots of copies of just few copies of DNA. I can copy different lengths of DNA, does not have to copy the whole length of DNA molecule. Okay, and the machine is called thermal cycler. What else we need? We need that machine. You need the sample which you want to amplify. You need enzyme DNA polymerase who is going to catalyze the reaction. Now that polymerase enzyme, these are the enzyme, right? So that means these are the protein. For thermal cycler, we really use high temperature, and protein, if we use protein in high temperature, they get denatured, right? So we cannot use our regular DNA polymerase. We collect this enzyme from thermophilic bacteria. There are bacteria who can tolerate high temperature in the hot spring, and all those, they are called thermophilic bacteria. So those bacteria, they use the tuck DNA polymerase enzyme. So inside their body, they have special type of polymerase enzyme. Those enzyme can tolerate really high temperature, all the way up to 120 degrees centigrade temperature they can tolerate. Okay, so they don't get denatured. So inside the thermal cycler, we use that DNA tag polymerase. We collect that enzyme from the thermophilic bacteria. We need DNA nucleotides, means adenosine ATP, adenine, cytosine, guanine, thymine nucleotides. That we have to supply, right? Pair of the primers. Remember, we must have primers, short fragments of RNA or DNA, because DNA polymerase cannot start if there is no small piece of DNA or RNA segment. So we must need to supply a primer, then water and the buffer. These are the requirements. So protocol put all reagents inside the PCR tube. Then there are 3 steps. Okay, one is called denaturation, means we have to separate out the DNA double strand. We actually increase the heat during this time. So these 2 strands get separated to each other. Then the annealing means each primer, so over here, each primer get attached with the ladder. And then the extension means DNA polymerase start making the DNA strand. Denaturation, it's 95 degrees centigrade, high temperature. Then priming or annealing, we then lower the temperature. This time, primers added in a concentration that favors the binding to the complementary strand of the test DNA. Okay, then extension, again we increase the temperature a little bit, 72 degrees centigrade. So polymerase enzyme start reading the sample, and it start making the complementary strands. Typically we use 20 to 40 cycles. So you can understand within 20 to 40 cycles we can make millions of copies of DNA, because each time from one DNA we make 2. So from 2 to 4, right? So after 20 to 40 cycles, we can make millions of copies of DNA, and it's a cycle. So basically, it goes on and on and on and on until you stop the process. So that's the PCR. That's the polymerase chain reaction. So what's the purpose? PCR can be applied in research, forensic, and clinical laboratories, determining the sequence of nucleotides in a specific region of DNA, amplifying a target region of DNA for cloning into a plasmid vector, identifying the source of DNA left at crime scenes, analyzing the sample to identify the paternity, find early signs of cancer. Okay, so basically there is a small tumor inside the colon. Okay, so we grab a very little bit of cell from there, and we can collect just few sample of DNA. Okay, for the research purpose. So from that cell, so we have to send in maybe 30 different research laboratory. They want this chromosome. So what we have to do? So we will we actually clone by using polymerase chain reaction, and then we can do the research. Find genetic defects in human embryos, same way. Examine the DNA of the ancient organism. Like we say, okay, so we found out a mummy, or under the eyes, we found out a teeth. Okay, so, and we can do the research. So from that teeth, we can extract the DNA, maybe millions years ago, that teeth came from a human, or from a cow, or from a monkey, from maybe 1,000 years ago. Okay, and we can make multiple copies, and then we can find out, that how long ago and how do they used to look and all those things. Recombinant DNA technology, what are the purpose? So mostly the pharmaceuticals, we make antibiotic drugs, vaccines, we make different types of hormone. That streptomyces, that's the bacteria, they make the antibiotic production. Yeast, by using yeast, we make that hepatitis B vaccine. Okay, we use E. coli for insulin and human growth hormone. I already talked about this slide. Okay, so we are done with this chapter. So do you have any question?
No, ma'am, I think I'm good. The only one left here.
Okay. Oh, you were the only one. Okay. Thank you, James.
Have a great night, Miss Nandi.
Yeah, you too. Bye.